think this is like our third year doing this, or second year, so it's getting like iteratively better. Yeah.
It's at least the fourth time I give this talk.
You've done it 4 times then? Then 4 times then, yeah. Yeah, okay. All right, welcome to the Simon's Electron Microscopy Center's Winter EM course. Today's lecturer is Dr. Tom Walz. Tom got his PhD in biophysics at the University of Basel, and he's currently a professor at the Rockefeller University, where he's using Cryo-EM to study membrane proteins within their lipidic environments. Today's lecture focuses on Cryo-EM map validation methods. As many of you know, since the inception of direct detector devices, the number of EM maps that are being deposited in the PDB and the EMDB is exponentially growing, and that just highlights the necessity and the importance of validating these maps. So we're really lucky to have Dr. Walz here today. This is an informal lecture, so please feel free to ask him questions, pick his brain. He has a quick search of his name. He has over almost 100 PDB structures and dozens of EMDB structures, so he's definitely one of the leading experts in the field. So without further ado, here's Dr. Walz.
All right, so my name is Tom, and I apologize again for being late. And so I kind of disagree with what Laura just said. Validation used to be really important when we were doing blobs and everybody got some kinds of blobs. So then it was really important to actually figure out whether the blob you see is correct or wrong. These days you typically get resolutions of at least 10 angstrom, usually better, mostly in atomic resolution structures. At that time you can build an atomic model, so validation of the map is kind of what you do as you build the model. So therefore, what I'm going to talk about is a little bit about validation at the very end, because if I just talk about validation, it's done in 5 minutes. So, but I thought what might be interesting to you is see a different guy, how he thinks about single particle electron microscopy. And this is really because this is towards the end of this lecture course. Ask questions if you have any questions and I come across these topics. Okay, so as Laura said, I'm supposed to talk about validation, as I feel it's not so important anymore. But it used to be that electron microscopy really had a dark side to it. And the dark side was something like, the really great thing about single particle electron microscopy is that no matter what kind of data you collect and no matter how you process it, you always really get a nice 3D map out of it. So the bad thing about electron microscopy is that every data set you collect and any way that you process it, you will get a density map. The big problem is at the beginning, about 5 to 6 years ago, we really had no idea whether the map that we generate is actually correct or total nonsense. And so that's why it was called blobology. We had a terrible reputation. And so this was really a big issue when we were working with these low resolution density maps. And a very nice example of this problem is shown here. These are 4 structures, all published within a 2-year span. It's all the same protein from the same organism. The only difference, it was done by 4 different groups and using slightly different methods and approaches. And the common saying is that at least 3 of these structures are incorrect. It turned out all 4 of them were incorrect. So that was really the problem. That was why it was really important that when you got these kind of maps, that you made sure that you have a correct map and not just blobs. And so I'm just going to take you through single particle EM, the workflow. You have seen this already. You have discussed all the different parts, but it will become clear why I talk about this. So the most important part is to actually get your protein from the cells. You have to express and purify the protein. And these days, that's really where your protein lives or dies. If you get a good protein that behaves nicely, it's almost guaranteed that you get a good structure. However, getting a good protein, that's a good old biochemistry, really important, really identifying whether you're going to get a good structure or not. So once you have the protein, you will prepare your specimen, you basically vitrify it. The reason is that your proteins consist mostly of water. So if you put it into vacuum, you get total dehydration, total collapse of the structure. So you have to prepare your specimen somehow for the vacuum. And obviously the current method that everybody uses, vitrification, either by plunge freezing or spotted on. But there's an old technique that many of you might not even know anymore, negative stain electron microscopy. It's a very useful technique. It's really funny, when I started doing electron microscopy, maybe about 20 years ago, 30 years ago, everybody was just about starting with Cryo-EM. And I was the throwback, and I got famous for getting structure with single particle negative stain EM. And now somehow history repeats itself. Everybody wants to do cryo, but before you do cryo, it's actually not a bad idea to look at your specimen by negative stain EM. It's a very good method to optimize your specimen. It's very easy and quick. So once you have your vitrified specimen, you always take images. So again, in the olden times, it was quite difficult because we had all this kind of specimen movement, and we had beam damage. So imaging was kind of a little bit of an art form, and so we ended up with very noisy images. And then what we have to do is we have to pick the particles, align them to each other, and classify them, so that we hopefully end up with structure homogeneous groups. From those we can calculate averages, and this is the so-called class averages. So the only thing that is different is you still have two-dimensional information, but you have a better signal-to-noise ratio. Once you have these 2D averages, the next step would be that you calculate your first initial 3D map, density map. That used to be, again, a fairly tricky endeavor, because you had to work with this very noisy data, and it wasn't so easy to actually get a reliable initial 3D model. But you tried your best, and you got your initial density map, and it's pretty crummy because you just basically guess what the rotation parameters are. So the next thing is then to improve your rotation parameters, which is called refinement. And more recently, we also did 3D classification to now also classify structures based on the 3D structure. And then we end up with a final 3D map, or 3D maps, depending if you do 3D classification. And once we have these maps, the next thing is obviously to either build models if you have high enough resolution. If you don't have high enough resolution, you take homology models or crystal structure of domains, and you try to dock them into your density map. So that's the typical workflow of single particle electron microscopy. So it starts out with molecular biology and biochemistry, which is very important. Then the electron microscopy, a big part of this is image processing, and then from the final map to getting the 3D atomic model is really just interpretation of your data, using chemistry to guide you. So what I'm supposed to talk about today is, once you get your final 3D map, how do you validate that this 3D map is correct? And that's different from another validation where you actually try to validate whether the atomic model is correct or not. Somebody else is going to talk about this. So I'm supposed to just talk about the final 3D map, how you validate it. My view is that at the point when you have your final 3D map, you should be already fairly sure that what you have is correct. And so my belief is that during the entire way, when you actually prepare your final density map, every step you try to optimize and to validate. So therefore, I'm going to talk about all these different steps and just give you my view how I think about things, which might be different for other people. And then at the end, I'm going to talk about 5 minutes about real validation of the density map, just to be consistent with the title. All right, so any questions so far? Everybody has heard this already 10 times, I guess, so bear with me. So the first thing, purify your proteins. And so if you have a protein, you put it on a grid, the issues that you might encounter are that you have very often heterogeneity. So the idea that you have a homogeneous protein preparation is almost illusory. Every protein population will have heterogeneity. The question is just how much of heterogeneity you will have. And the more easy one that you have to deal with is compositional heterogeneity. If things fall off, it's quite easy to classify. Conformational heterogeneity used to be a real problem, and to this day is a problem if you have continuous movements. If you have a domain that just moves around, it's going to be very difficult to actually get a really nice structure of this particular domain. But discrete conformational heterogeneity and compositional heterogeneity are pretty much problems of the past. These have really been sorted out by now. Another thing that I actually want to talk about more specifically is the effect of cross-linking. A lot of people use graphics, which is really a great method, or on-column cross-linking, or just in-bulk cross-linking. It's a very useful thing to do, but the thing is, when you do it, you have to think about what you're doing, and you have to make sure that it doesn't affect the structure of your protein. And I will give you an example about this. So my honest belief is any sample that you ever put into a cryo-electron microscope, look at it by negative stain electron microscopy. It takes you literally 5 minutes to make a grid, put in the microscope, and see immediately what you have. So whenever you have a nice band on your gel and you have a nice peak on a gel filtration column, it means nothing. It's kind of like mass spec. Negative stain electron microscopy sees everything. And so even if you have a nice band and a nice peak on a gel filtration, your protein can still have all kinds of different conformations. So it's a good idea to actually look at this by negative stain electron microscopy. Just making a quick grid is very simple, take some images. And the reason negative stain EM is so useful is because you absorb your protein on a carbon film. And so you actually minimize the orientation it will absorb. If you lock it's just one orientation, sometimes you have a few orientations, but it's not the random orientations that you have in Cryo-EM. So if you have two particles that look similar but somehow different, it's not different orientation. You know immediately you have a conformational difference. So ideally, you do everything you can in the biochemistry world to try to minimize your heterogeneity, especially if you have moving part, if you can find a substrate or an inhibitor that actually locks it in a particular structure, it's going to make your initial first structure much easier to get. So you look by negative stain, which increases your contrast, nails down the orientations, you see whether you have heterogeneity. If you do have heterogeneity, and you're not quite sure how bad it is, these days it's very simple to just take a few hundred images, negative stain, automatically pick the particles and calculate class averages. It might take you a few hours, but then you have a really good understanding of what your particle is, how heterogeneous it is, whether it's going to be a problem or whether it's a minor problem, you can ignore it. So I always recommend test your sample with negative staining, optimize your sample before you go and do Cryo-EM. And so if you need chemical fixations to prevent your particle from falling apart, that's a totally fine method, there's no issue with that. But you should make sure that when you cross-link, it doesn't change the structure of your protein. And so a nice example is shown here by Yorgos Kiniotis' group. This was a GPCR that he had a complex with beta-arrestin and a little antibody, a Fab fragment, just to make it better visible. And so you can clearly see the GPCR. And then here, this is the beta-arrestin. And if you don't cross-link, you can see that the beta-arrestin is in two different orientations. Either just barely hangs on, just with a small little contact here, but it basically dangles around, or it engages fully, and you have a really nice interaction between the GPCR and the beta-arrestin. So if you don't cross-link, you can actually see this initial engagement step, and then later the final docking of the proteins. But if you do cross-linking, and this was actually done with this new method, which is called on-column cross-linking, which works very nicely. With this one, he could only find this really fully engaged beta-arrestin, and this conformation state, or this initial engagement state, was completely missed from this. So there's no problem to do the cross-linking, and you will get the structure of one of these states, but you might miss out on other states. And so you might want, whenever you do cross-linking, not a bad idea to have a quick look at a non-cross-linked sample. It's going to be much more messy, but at least some particles should look the same as the particles that how they look after cross-linking. And so here you have at least some particles that look the same, but this is another example that is much worse.
Is that, so those averages from that
Are you a student? Are you a student?
I totally am a student of life. That image before, those are all from negative stain?
All negative stain.
Perfect, okay. Yeah.
Why
That's
not?
it. No, I just wanted to know like what
The resolution is too high?
know what to expect, right, for 2D class averages versus in cryo versus negative stain. So
Yeah, so cryo you
yeah.
see much more,
Yeah.
of course, but
Okay.
there's also a small particle. And so this, for me, is still sample characterization, is not structure determination. You just want to
Okay,
know what the
sure.
structure actually does. And so before you go into cryo with cross-linked samples, check a negative stain whether the sample look the same before and after cross-linking, whether you just stabilize the structure and don't do anything else. And so that's also negative stain structure. So this is Hobbs complex. This was done, again, this was actually done by graphics. And it looks very nice. It has these two blobs. So you can actually get very nice 2D averages, and we're also able to get a three-dimensional reconstruction of this. And I was working on the same complex at the same time, and I was very frustrated about this. And the reason is that our structures look very, very different. Our structures look very, very messy. So you have a lot of stuff that breaks apart, and it's really. But the reason is that actually what the structure looks like is that you have this little head domain, and then you have 3 legs, and these 3 legs can go in all kinds of different orientations. And the legs can actually fall off, or the head can fall off, everything can fall off. So cross-linking would not be a bad idea to do, but what you do is you basically stabilize the head, and then you clump all the legs together, and so now you have a domain for the head, and basically all the legs clumped together. So you can get a structure from this, but it has nothing to do with the structure of the actual protein in solution. So therefore, this is an extreme example, but it's a good idea if you do cross-linking, that you see that the cross-linking doesn't really affect the big arrangement of the structure.
I have a question.
Yeah.
Do you get aggregation problems when you cross-link it from when like 2 molecules might cross-link together, or?
You can. So the trick is to optimize the cross-linking. How much? So if you do it in batch, you have to optimize the cross-link concentration, glutarate concentration. But that's why these new methods were developed, graphics and on-column cross-linking. And all the methods, graphic is by far the best. And what you do is you actually spin the protein into a glycerol gradient, but at the same time you spin it into a fixative gradient. And so as they go into the gradient, they get fixed. And so you actually fix it and then isolate a nice complex. And so you get a very, very nice fixed sample. The problem is it actually becomes quite dilute, and so you have to concentrate the sample again. But graphics is by far on the way from all the techniques that I have tried really the best every single time. And there you don't have to optimize the fixative concentration because you have a gradient already. So it does whatever it needs to do whenever it needs to do it. So graphics is highly recommended. We use graphics a lot, but we always make sure that it doesn't change the structure of the complex. So that's the protein, and when you have the protein, obviously what you need to do is you have to prepare it for the for the electron microscope. And so you can have all kinds of issues, and the common one is that you have beautiful particles, negative stain. You make a vitrified grid, and there's no particle. All the particles are gone. Happens quite often. Another problem is that you get nice particles in your host, but they are all in the same orientation. And the problem with that is that you have to see different orientations so you can actually construct a 3-dimensional density map. So you need the different views. So these are the 2 different problems. No particles is not so much of a problem anymore. People now have found all kinds of different ways to deal with this problem. Often you just have to increase the protein concentration. Another method is that you apply the specimen, you blot, and then you apply the specimen again. And the idea why you don't see the proteins, the holes, is the idea is that the protein actually go on the surrounding carbon film. And so when you apply the protein for the first time, you cover all the carbon film. That is now saturated, and if you now apply the sample for a second time, the idea is it will now go into hole. So that works sometimes. Another method that is not very frequently used, but you can basically just coat your carbon film with PEG, which is a hydrophobic brush, so the protein doesn't absorb to there and it pushes the particles in the hole. You can use different grids. Gold grids have been shown to behave differently from carbon or copper grids. Lacy carbon, for whatever reason, sometimes works better than carbon grids. And then you can, of course, use a thin carbon film or a thin what's the name? Graphene oxide, to help to absorb the particles into the hole. The problem with carbon film is it will increase the background a little bit. Graphene does much less of that, but graphene is more difficult to make with grids, at least for us. So using one of these techniques usually will solve your problem, and you will end up having your particles in the hole. But a problem that is more tedious is the preferred orientation, and so this is all due to this guy. So the problem, that's your fault.
It is your fault.
Without you, it would not happen. So basically the idea is if you have particles and you make a very thin layer of your buffer, you have a fairly high chance that your particle is going to get into contact with this air-water surface, air-water interface. It will do this actually hundreds of thousands of times. And because of this, of the surface tension, particles tend to get stuck on the air-water interface. And they start to align kind of when you throw a coin on the floor, it will always lie face down. Kind of same principle with particles on the air-water interface. So that has been a problem for a long time, and people are still struggling with that. And the problem if you have this kind of issue is that you might end up having certain orientations that you don't see. And so if you don't see certain orientations, these areas or these directions of your map are not defined. And so you can actually create incorrect maps. So this is about 10 years ago, we were working on the Tor complex. And that was exactly this problem. In this case, we could get very little of this protein. It was very difficult at the time to purify it, so we had to use a carbon film to get the particles in the hole. But then, when we calculated averages, literally every single average showed the complex in the same orientation. So that was an issue. And so what happens is that the view that you have on your grid, you can actually nicely see the different features. But perpendicular to this, there's virtually no distinction. It's just one. So you could almost just take your 2D average and spread it out, and you would have the same structure. So that was a problem. But there are again all kinds of different ways, and these are just few of the ways that you can fix the problem. You can use thinner or thicker ice. Thinner ice might damage your particle, thicker ice might create too much background, but sometimes it works. What is most often done is you use a low concentration of detergent, which will change the surface tension and will minimize the effect of particles trying to align to the air-water interface. You can use a thin carbon film, and that has been done extensively for ribosomes. You can use gold grids. So these are all kind of ways to try to get different orientations. But the real hope for everybody is that you can actually now do spotted on. And spotted on seems to, in many cases, avoid or minimize the problem of preferred orientations. If you can't avoid it, you can use tomography or you can use tilted data to get the different views. But it's rather painful, the tilted data, although it has been shown here that you can get to very high resolution. But it's even in this place where it was developed, nobody seems to ever do it. Is this correct?
How many people do tilted?
It's
Percent.
of people who have preferred orientation, probably at least that might be the first thing they might try to do if they have Cryo's time.
Mm-hmm.
I have found that I'm probably working with at least 1 or 2 users per month who want to do a tilted data collection. Usually what ends up happening though is they want to go back to the bench and just fix it biochemically in some way. But
Why?
Yeah.
Why?
Why do they want to fix it?
Because the paper says you can get to high resolution. You got the structure at 3 angstrom. Why is this not a good solution?
It hasn't been so easy. The throughput of it is just not very great. So so in my experience, one, you start to get thicker ice. You're literally limited to using gold grids because you have to tilt your grid, so you're going to have a lot of drift. The defocus gradient, there's a lot of different methods to estimate the defocus across that inherent defocus gradient. It's not entirely sure what is the best way to do it or how accurate it's measuring it. There's so I don't know. I don't think it's it's really robust yet. But I do think with test specimen, it's been pushed. But with actual biological, maybe there's HA. But my particular protein, the tilting never really got any better than 4.5 angstrom.
Oh,
yeah,
that's horrible.
I know. They're telling me. I was telling the collaborator it looks great, but in whatever it is, it's
4.5 is a lot better than nothing.
I agree. I don't know what. I agree.
So I actually tried tilting, and we had a very strong preferred orientation, and I think that the TM domain was actually aligning on the interface and denaturing, because we couldn't get anything with tilting, and then adding detergent completely fixed it.
Yeah, so sometimes people just add an additive like detergent. They just want to buffer up that air-water interface, tuck the protein in, and that seems to help sometimes. Obviously, with membrane proteins that are already in detergent, you don't. That shouldn't help
Well, it
because
was an amplifier, actually.
sure. But if there is already detergent in the buffer,
have that,
yeah.
Yeah, so that's a little bit surprising to me. If you have a membrane protein, it should be in detergent to begin with. Why do you need to add?
We went to amphibol.
Yeah, so there was no detergent in the buffer solution, right?
We
So
just had like
yeah.
a small.
And it didn't mess up. Amphibol doesn't.
It seems
matter.
okay. Yeah.
Yeah.
Okay.
That was like the same thing when we were trying to play with beta gal to do like tests, get the Cryo-EM, all the specs going. And with beta gal, we also had a lot of preferred orientation, but in the paper they add like NP40 or something or OG like below the CMC, and then like preferred orientation just goes away. I mean, you have other things like daisy chaining and stuff, but then you get both views. So
How do you get rid of daisy chaining?
that's a great question. It's just random for us sometimes.
Okay.
I cannot explain why. Yeah. So, a lot of this is just if you get a good grid, you just collect on it, and we don't ask why anymore.
So, but you have samples that do daisy chaining. You do occasionally have samples that don't.
Yeah,
The
off
same
the
brand.
same tube. Yeah. Yeah.
I just think it's
So you're now responsible for that. Anyway,
Is
so
ice thickness I mean, it kind of varies, but yeah, you'll see it kind of just make these little stacks and looks like a little snake of. Yeah.
So I basically agree. You try everything else before you start tilting, despite your comment.
Yeah.
But I'm now at the point where I will start tilting because we can't fix it any other way.
I think it's kind of the same argument with like faceplate data collection.
Yeah.
Like, do faceplate last? Like, don't make that your first go-to because it's just not super robust. But it's still a good option.
Sometimes.
Sometimes, yeah. As is tilting. Sometimes, yeah.
Okay. So that's another example. So this is kind of what you have. This is PAX 16 and AAA ATPAs. Looks very nice in the images. You calculate averages. Every single one shows you the triangular top view. So then you add some detergent. It starts to look actually quite horrible in my opinion, but you end up with all kinds of different orientations, and that allowed us then to get a three-dimensional density map. So, yeah.
Ours looks horrible with DDM, but
It looks horrible, but
Yeah,
then
it
it gives
works.
good, yeah, so you take it. All right, so then you have your grid, you do the imaging. And so again, in olden times, there was an issue. And so the potential issue is that you had low contrast, and you had to really be worried about beam damage. And so the basic idea is, when you vitrify a specimen, all the contrast that you get comes from scattering of electrons on the specimen. And the idea is the bigger the atom is in the specimen, the more scattering you get. The scattering is roughly proportional to the atomic number of the atoms in the sample. Now, unfortunately, biological specimens have carbon, nitrogen, oxygen, hydrogen, all very small atoms. So inherently, biological specimens are not very good electron scatterers. And so it's not just the protein, but also the buffer and the salts in the buffer, everything will scatter. So the difference that you see as contrast is really between what the atoms in your sample scatter versus what the atoms in the buffer scatter. And that is not very much. So you end up inherently with a poor contrast. So to avoid this or to overcome this, the best solution would be just use more electrons, you get more scattering, you get more signal. Of course, you cannot do that because your specimens are also beam sensitive, so if you use too many electrons, you break bonds, the fragments diffuse away, you get total structural collapse. So that's not a very good solution. So that was always the dilemma in single particle reactor microscopy. We want to use a lot of electrons to improve the signal, but we can't because we create damage. So the poor signal to noise ratio you can actually overcome by doing averaging. You have to just collect a lot of data of a lot of particles in the same orientation, and then you can average and you can computationally boost the signal to noise ratio. So that you can fix, but once you have beam damage, you cannot fix that. If the sample is broken, it's broken. So therefore, what people used to do is to use low electrons to take an image. The particles were hard to see, and then you were trying to pick them and to classify them, get averages. All this problem obviously has been solved a few years ago with these direct electron detector cameras. Have people already talked about this? They must have.
Yeah.
So But they have been really amazing because so first of all, the signal-to-noise ratio, the MTF, the DQE, whatever you want to call it, is much better than with a CCD camera on image. But not just that, the really big advantage is that we can collect these movies, and that really has changed the field. And the reason is we used to just take a single image because we couldn't collect movies. The problem is at the moment the electron beam hits your specimen, you deposit a lot of energy on your specimen, you have energy that needs to dissipate, and so the specimen has to start moving. It always starts moving. And so, because we only had one shot at it, all our images were blurred just because of the initial hit of this. There was nothing we could do about it. So with movies, obviously, you can now align the individual frames and computationally correct the movement and then average together the frames. And so you have much better images. You can basically get rid of the blurring of the images. In addition, you can now take many larger movies, much larger movies, and what you do is you do resolution filtering. So at the beginning you take all the information, and as you continue in time and you get more and more beam damage, you just take less and less of the resolution in the later frames. The great thing is that all the high resolution information that you have in your images are from the initial frames, which have little beam damage, so they're accurate, but you keep all the low resolution information on all the frames, and that helps you very much to align the particles, to classify the particles, because a lot of the information that you use to do this is actually in the low resolution range. So these are really amazing data that we now can collect, and so that's where now everybody can actually collect high resolution data. It's really has become almost trivial these days. But it used to be a problem. And so imaging is probably now almost the easiest part of the whole project. It's pretty much standard these days, how you do it. But then you have to pick the particles and align the particles and classify them just basically to boost the signal-to-noise ratio. And there are problems that can and have occurred. And one problem is modular reference bias, and that has been a fairly serious problem. And then also when you do the 2D classification, again you have model bias. And then it's very difficult if you do 2D classification, how many classes do you actually want to calculate? Are your classes homogeneous, or are they still pretty heterogeneous? And then there were some classes that disappear completely. So who of you actually uses Reliant?
Not yet, I will.
You will. Okay, who is or is planning to use Reliant? Reliant is horrible with 2D classification. It's the worst program ever I've seen with 2D classification. The good thing, it doesn't matter anymore. So, but you can actually gain a lot of information from 2D classification, just not with Reliant.
Can you go more into depth about why Reliant is not good for 2D?
Can I go 2 or 3 slides further?
Sure, yeah, yeah.
We'll do that. So first, the problem with the reference bias. And so there's a very famous case. This is a noise image. Have you seen this already in the course? Okay. You have seen the Einstein. He has lined 1,000 images, so you create Einstein from noise. So that actually used to be a real problem, because this is a paper that was finally published at about 8 angstrom. But in the beginning, this group wanted to publish a structure at 1.9 angstrom or 1.8 angstrom with an F20. It was actually fine. So in any case, this was what the images looked like, and then they got these amazing averages where you saw all these details. And then they got 8 angstrom out of it. And that created a big, so everybody's laughing. This had a big stir in the field, and some of the senior guys tried to explain. So a lot of us tried to explain to these people why they shouldn't do that, but they wouldn't listen. So in the end, they published it, and then several people sent responses. And this is the response from Richard Henderson. He just took 6 images, averaged them together. So these are just 6 of these single particle images, averaged them together, calculated, and then just changed the contrast. And what you see is this bright ring, which is probably a particle, and then it becomes very dark, and then it becomes bright again. That should not happen if you just have 6 images that you average together. So what Richard could deduce from this is that they actually used a high-resolution model to pick the particles, and that's where they got the information from in the end. And the other problem is, this is the image that they collected, where you can barely see the particles, and probably most of it is just simple noise. And a protein of this size, if you take a real image, should have about this contrast. So they just had really bad data and did horrible image processing and got 1.9 angstrom. So anyway, these days you actually can get 1.8 angstrom, but in a very different way from what these people did. So the take-home message is, it's painful to pick particles, but the first few images you really should pick by hand and really calculate averages with particles that you pick for two reasons. First of all, if you stare at your images, you can actually see the quality of your particles and you get an idea how heterogeneous you are. If you let the computer do everything, you start to forget what your sample actually is. So it's good for you to spend some time with your sample, look at the particles. And then you calculate your averages, and those averages are fairly serious because it was interactively done. And so you can then use those averages to pick the particles automatically. But if you start with auto-picking with templates that you choose from wherever, the particles will end up looking like the particles that you used, the reference that you used to pick. So it's a good idea to really first pick some images by hand and calculate your own averages. So k-means classification is the typical way that the people calculate 2D classes. And so the two different ways, so I'm not going to talk about k-means, but there are basically two ways. In k-means classification, you have to give the program a number of classes that you want to get. And so how do you get these classes? And so how do you provide the references for that? And so one way is that you just give a number of templates and you say, tell the program just classify into these classes. And so this is also called supervised classification or multi-reference classification. That's deterministic because you tell the program this is what the particles look like, classify into these. Now, the problem with this is again that you have reference bias because you give the references, and so the program will do its best to put it there. So if you give that templates, you will get some averages that look like your better templates. So rather than the deterministic initialization, you can do random initialization where you don't give the references. And you basically just tell the program, okay, I want to have 100 classes, and then the program goes and randomly selects 100 raw images, and then uses this as a beginning. Alternatively, if you tell the program, you can tell the program I want to have 100 classes, and the program randomly distributes all the particles into 100 classes, calculates averages, and then uses those averages as the initial templates. They're going to look horrible, but there will be tiny differences, and so that's a fairly unbiased way to start a classification. So that's a better way to start. The problem is it's an unstable approach because if you restart the program and the program uses 100 different averages to begin with, or 100 different images, you likely end up with different results. So it's not a stable way. Not every time you run the program, you will not always get the same averages out of it. But these are the two ways that k-means work, or you initialize it, and then you should know a few properties and an issue with k-means classification that people tend to forget.
Question, the templates, the first method, where do those templates come from? You just add
So these, if you, for example, have different data sets where you have, well, let's say you have a proteasome, and now you want to have a proteasome with something bound to it. So you just take the proteasome, get some reprojection from the proteasome, and use those as references. Or you get it from crystals, wherever you want to get them from. You can also basically just give random shapes, but people usually don't do that. The typical way is that you get, you know kind of what your protein is going to look like, and you take references that represent that. But this is really the way people usually do it. So actually, we actually usually use this. We just select 100 images, and we just deal with the fact that it's not stable. So we can do it a few times, and then we know which averages are actually meaningful and which are not. But I will show you what we use, and which I really like, but nobody uses it. So something you need to know about k-means is it will always converge. You will always get averages, but it's not a global minimum. So you can often get into sub-minima, and so sometimes you actually miss some classes because they just didn't come out. So this doesn't always give you the full description of your data set. It's a problem if you have outliers, if you have few particles that are quite different. They will really very badly affect your k-means classification. That's just an inherent problem with k-means classification. And then another problem is that some groups can collapse. If there are very few members of these groups, and you have a group that is very strong, it becomes better and better in signal-to-noise ratio. So these particles will get sucked out of these groups into the bigger groups. And again, you lose views of some of the smaller classes that are in your data set, but you will never see them, especially Reliant is really horrible with that. So you have, say, 100,000 particles, and in the end you get 60 averages. There's just no way that this really represents your data set. But it doesn't matter anymore because these days everybody uses 2D averages just to get rid of junk. But you can actually use 2D classification to really understand your sample. And so the problem is that you have to guess how many classes you want to have. And if you have perfect particles, that's not a big issue. But if you have noisy data, it can become really difficult to actually judge whether differences between two classes are meaningful or just different noise. So this is long known problem for 2D classification. And so Paul Penchak developed a very new technique which he called ISAC for iterative stable alignment clustering. I really love this. It's an extremely powerful method. So what it does is that it doesn't allow a class to grow as big as it wants. It restricts the class. You actually basically tell the class each class cannot be more than 100 particles. And so even if you have a small class, it cannot be sucked into a big class because there's a limit to how many particles these big classes can have. So this really means you will not lose your minor images. And then what it does, it basically does an alignment, classifies this as an alignment, and then it does this several times, and it checks whether the particles in this class, the alignment parameters are stable or moving around. If they're moving around, it means this is not a very good particle because it's not easy to align or classify. So only the ones that stable, always align in the same way, will be considered as stable particles or stable classes. And then when it has the stable classes, it does the whole thing once again several times and checks whether the same classes come back if you do the same thing several times. So that means in the end, if you have averages, they're both stable and they're reproducible. So they're very reliable. And that means your classes are very homogeneous, and this used to be very useful when at the times when you use the 2D averages to calculate your initial 3D models. So if you get these 2D averages with ISAC, you could really believe that these are meaningful averages. Now, it has obviously drawbacks. Once it calculates these classifications over and over and over and over and over again. So if you have a large data set, it can take a week to calculate this stuff. It's just very, very slow. And also, if you have maybe 100,000 particles, the first iteration of this can be that only about 2,000 particles are selected, depending on how stringent you are. So you get very few particles, but then you can run more iteration, you get more and more averages. So it's slow, you get very few averages, but you will see all kinds of different views, and they will have very good signal-to-noise ratio because they're going to be very homogeneous classes. So I really love ISAC, especially if you do negative staining. You can get a lot of information out of this. The problem is ISAC these days suck. ISAC is now implemented in Sphere, this program, and the ISAC that has been implemented in Sphere doesn't do anything as it should do. So if you now run ISAC on Sphere, you get almost all the particles are assigned to classes and the classes are very blurry and clearly not homogeneous. So if you want to run Isaac, do not run it in Spire. Try to find an old version that is distributed with eman2 and spokes. It's very painful to install it, it's very painful to get to run it, but once you have it installed in your lab, it's really, really powerful. It's a really great program. So now you have your clean 2D averages. Is this what you asked? What did you ask?
I was asking why you don't like Reliant. You answered that though.
Yeah. You get group collapse. I find it astonishing if you have 1 million particles in a data set and you get 50 averages, which show very few different orientations. So it's really a horrible way to do it. But because you only use these days Reliant to get rid of junk classes, it doesn't matter anymore. Okay, so now you have 2D averages, and you have to do a 3D reconstruction. And the issues are that you might get an incorrect map, and that was because you basically have 2D projections, and you have to try to figure out how these projections are arranged in 3D space. And that's actually an underdefined problem, and used to be a real problem at the beginning. And so you can get an incorrect map because you have a heterogeneous sample and you didn't do a good job separating the different particles. So if you combine all of them into a single map, you get a mishmash of all the different structures. You can have missing views, and so you get areas of the map that are not defined, and simply the program can make incorrect solutions, especially in olden times when we used the magic and things like that. Getting a real structure out of this kind of thing was almost impossible, to be honest. I mean, that's why so many structures came out wrong. But again, all this has gone away. It's become much easier. But just to say how we used to do it when it was still a problem, we used to do random conical tilt, which has now come back to some degree. But the idea is that you have, usually when you do random conical tilt, you use a carbon support, and so you have a preferred orientation. So you know the orientation of your particle. And so you take an image and you get a projection image and you know what the orientation is of that. But now you can tilt to 60 degrees, and you take an image of these tilted images, and now you get different views and you get different projections of your molecule, of your sample. And so what you do is you align the orientation parameters to these different tilted views, and all you need is you have to define the in-plane rotation of this molecule, which is very easy to get. And then the second orientation you get because you know that you tilted by, say, 60 degrees. So this is geometrically very clearly defined, so you know every projection what the orientation is. So now you can assign these orientation parameters to the projections, you put them into space, and then you just simply shine light bundles through these projections, and they will carve out a three-dimensional reconstruction of your density. And so this is a very, very accurate reconstruction of your specimen. It has some problems like missing cone. If you do negative staining, you have all the negative staining artifacts. But at least geometrically, this is an absolutely defined problem, very easy to get a 3D reconstruction that you can trust. But it's painful because you have to tilt, you have to pick two particles, one from tilted, one from untilted, so people didn't like to do that. And also in ice, in vitrified ice, you usually don't have the preferred orientation, so this approach doesn't work anymore. So if you have vitrified specimen, you have your particles, that's an old image before his paper, where the particles in all kinds of different orientations. Now we know that all the particles are actually on one of the sides on the surface. I will have to update my slide. So, but in any case, you have all kinds of different orientations, and if you take an image from the top, now you get all kinds of different projections from your particles. And what you now have to do is to assign orientation parameters to these projections. So you basically have two XY coordinates, where is your particle in your image, and then you have three Euler angles, and when you can decide on these geometric parameters, you can put it back into space and then reconstruct your 3D density map. And one of the ways that this used to be done was a method called angular reconstitution, which is complicated, but basically just had to choose three projections which you believe were perpendicular to each other. And you can see already where the problem is, because you have no clue, if you don't have the structure of the protein, what are 3 perpendicular views of your protein. So that was a big problem, but then you can, once you have the anchor set, you could try to find common lines, and you started to fill in all your other projections. And this was the sinograms and how this was done, it doesn't matter. But the problem is, it was a very iffy procedure and often actually gave you wrong results. And so what we do these days is actually based on an improvement of refinement. So the initial model is actually not nearly as important as it used to be. So this is just a few 2D projections that we got from this potassium channel, and we got an initial model. And you will see that the initial model has no details whatsoever, but you don't care, because it's just the initial model, and you now can refine all your particles against it. However, if this model has bad features in it, you will artificially increase these bad features through the refinement procedure, or you used to. And so that's why it used to be very important that the initial model was correct, so that you wouldn't reinforce wrong features. And so the way this was solved was initially a program called Simple, and it was a very simple advance, and this is stochastic hill climbing. That totally changed the world for single particle electron microscopy. So this has to do with refinement and how we used to do refinement. You have your initial density map, and now from this density map you calculate a projection of all kinds of different orientations, and so you now end up with a stack of reference projections from your initial density map. Now you have these reference projections and you have the experimental projections. So this is just one, but you have thousands of these obviously. So now you take one of these experimental projections and you compare this with all your reference projections by cross-correlation. And what you end up with are these cross-correlation peaks. And so the idea is the higher the peak, the more similar this experimental projection is to this reference projection. So the one that has the highest correlation peak is going to be the closest reference to this experimental projection. And because you have calculated these reference projections, you know what the orientation parameters are. So you can now take these reference projection parameters, assign it to this experimental projection, and so you have essentially improved your orientation parameters. So you do this with all your different experimental projections, and then you put them into space again, you calculate a reprojection, you get a new density map, you use this density map as your new reference, and on you go. So there's an iterative improvement of orientation parameters. The problem with this is, this will define your refinement. If this thing is wrong, it will find the best correlation to your wrong structure and will improve your wrong structure. So that's why it used to be so important that this initial map was really good. So this was called steepest descent, this refinement procedure, basically finding the highest correlating density reference projection. Now what has really revolutionized EM is a very simple idea. And this is, we don't do steepest descent, we just do stochastic hill climbing. What this means, you do exactly the same, you calculate all your correlations, and then you check the first one, and it's bad. You check the second one, it's better. And then you check the third one is actually worse than the second one. And so you say, okay, I don't care what is below here, this one is better than this, so I take this one. This is now going to be my best correlation. What I will use as orientation parameters, I assign it to this, and the rest is exactly the same. What is different is because you're not looking for the best correlation, you just look at the first best correlation. And so that might actually be quite different from what you started out with. And this actually allows you, the reference map, to emerge and to evolve and to become different from what it used to be. It's a very small difference, but it really totally changed the field. And with this, it's now actually quite simple. You can start almost with an egg, and using this procedure, you can actually carve out the real structure out of your initial data set. So this has been very, very important. And so this has initially introduced by Simple, and then it was used as Viper in Sparks. And now CryoSpark is a program that does it very nicely, and is Reliant doing it as well. Probably Reliant is doing the same thing. So this is now basically how refinement is done using stochastic hill climbing. So now you have your initial 3D map, and now you have to do 3D classification and refinement. I just talked about refinement, the same issue. You have reference bias, so if your model is bad, you will overfit. And a very contentious area, topic, stupidly contentious in the field, is resolution assessment. And so I'm not going to talk about this, the same idea that you just have over refinement. So resolution is a big area forever in single polygon electron microscopy. And so the way that it's done is you have two maps, and you calculate the Fourier shell correlation, which is done in this way. And if you do this in a resolution-dependent way, you basically get a curve, how similar these two maps are at what resolution. So this curve, it's known how to calculate it, it's quite easy to do it. It can be influenced depending on how tightly you mask, but basically you end up with a curve. Now the question is, where in this curve is your resolution? And what used to be the case was, okay, let's say when the noise is as big as the signal, that's cut off, that's the resolution. And so this was introduced by Bettina Böttcher and Tony Crowther. This is the FSC equals 0.5 criterion, which was used for a long time, and so you get one value. But this is arbitrary. Why is your resolution when your noise and your signal is the same? So that was really an arbitrary at the beginning, 1997, 20 years ago. That's what people thought about. Then later, Richard Henderson said, well, there's a better way to do this. And the idea is we use the same criterion as we use in X-ray crystallography, where 60 degree is random. A phase error of 60 degree from that on is random. So let's say this is the map is actually dominated by noise, and that correlates to Fourier shell correlation of 0.143, very famous number. So this was the new resolution criterion, and suddenly all the maps had much better resolution. But there was a big problem with that, and the problem is that this value only works if the two maps that you compare are totally independent of each other. And this is not what happened. People have never calculated independent maps. So what people really have done is they started with a stack of particles. They had a reference map, and they refined all the images against this reference and assigned new orientation parameters to your images. You created a new refined map, and this became your new reference map, and you kept going. So all the images were always refined against the same. And then in the very last step, you took your data set and you split it into half, and then you compared them. And you got two half maps, and you did Fourier shell correlation. And the problem is that these two half maps are not independent because they were all aligned to the same reference. So to make them independent, what people now do, and this is called the gold standard, which is absolutely stupid. There's no gold standard. So that's one of my pet peeves. I hate gold standard. And I will explain why. So you start with your particle stack, you split them in half, and now you take reference maps, and that is actually the same reference map that you use. So it's still not independent. You still refine it to the same reference map. But now you get these new orientation parameters, you get refined maps, you go back to the reference, and now supposedly you have two independent half stacks. So now you can gold standard compare those because supposedly they never saw each other, which is incorrect because the reference is the same. So there's no gold standard. And so the point that I want to make is that even if you would do it this way, you can still get the correct resolution. So you do the same thing, you do the Fourier shell correlation, and you can still actually, you can still use the 0.143 criterion, and you can still get the right resolution. The only thing that you have to do, you can only use the sub-region of the density map to refine. So if you compare and align your structure to a reference and only allow information up to say 8 angstrom to be used, or 10 angstrom to be used for the refinement, all the resolution from 10 angstrom to 8.7 angstrom has never seen the reference, has never been biased towards the reference. So this is totally independent resolution. And this will give you exactly the correct resolution as much as the gold standard. So all this nonsense about gold standards is a little bit annoying to me, but it's certainly a valid way to calculate the resolution, but there are other ways to calculate a valid resolution. And whether this is really the correct way to cut off the FSC is still up for grabs. People can still discuss this. Isn't just in the moment the accepted value. This could change again. So it's a valid, but even a gold standard FSC can give overestimated resolution, and depending on how it's implemented, it actually will give you overestimated resolutions. And the point that I really want to make is don't get hung up on resolutions. Resolutions really don't matter. Resolution is just a number. It's a useful number because it helps you to improve your density map. But whether you have 4.2 or 3.8, in most cases really doesn't matter all that much. Because it's just the resolution of the overall density map. And as most of the people who have worked actually on density maps, different regions of a density map will have very different resolutions. So it doesn't matter what the overall resolution is, just a number. Local resolution is much more important to understand your structure. And again, even local resolution is just a number. What really matters is what you actually resolve, and that's what matters. So a little test, if you have a 20 angstrom resolution, what do you see?
Smooth.
What is the technical term of a blob?
Surface.
Domain is good, what?
Surface.
Let's call it a molecular envelope. Or a blob. Anyway, so 20 angstrom, you get a protein envelope, and you can resolve domains and things like that. 9 to 10 angstrom, what do you expect to see?
Tubes for alpha.
Excellent. If you claim 10 angstrom and you don't see alpha helices, unless it's a beta metal, you are going to be an issue. Yeah. If you claim better than 10 angstrom, you better show some helices. At 4.8 angstrom, what do you expect to see?
Pitch.
Hmm?
The pitch.
You will start to see the pitch. What else is at 4.8 angstrom?
Strands.
Hmm?
That's strands.
You start to resolve strands in the beta sheet, so you can resolve beta strands. At 4 angstrom, glycine.
Not glycine.
So which ones?
The fat ones, tryptophan,
Yeah.
tyrosine.
The bulky aromatic side chains. That's what you're going to see. And so, what is really different in an EM structure and an X-ray structure is the 4 angstrom map in EM is really beautiful. The 4 angstrom map in X-ray is horrible. So, because we get the phases directly, we don't have to get them indirectly. So, we can actually get good models at 4 angstrom. You can't register everything normally, but you get fairly reliable maps, models at 4 angstrom. And that's just what is shown here. These are some, at 20 angstrom, you get the individual subunits of the virus. 7 angstrom, you start to see the alpha helices. Around 3 angstroms, you start to see the aromatic residues. And then at 2.6 angstrom, you see everything. So that's important. If you want to publish a map, make sure that you see what you actually claim that you can see.
I'm sorry to interrupt you. We have about 8 minutes before the class ends.
Okay.
But we can definitely extend this like maybe 5 to 10 minutes if the students are okay with that.
Yep.
Because I know you're kind of like 3/4 through your lecture.
This is very quick.
Okay, yeah.
This is validation.
Okay. All right, continue please.
So the idea is if you do everything correctly these days, validation is really not necessary anymore because you usually get to high enough resolution that you will see alpha helices, you will see the pitch, so you know the handedness and all this. But in olden times, this was actually not the case. You get these kind of structures. How do you decide which one is correct? And so in 2012, the field came together to have a review meeting to say, what do we expect to see when people publish density maps? And so they came up with a list of things that they want to see for structures. And so one of the things was you want to compare reference-free averages with projections from your density map. And so this is one of ours. At the time, we had anaphase promoting complex, and we always compared raw images with reprojections and class averages, and they were supposed to look the same or similar. this does actually not tell you that your map is correct. All it tells you is that your 2D average is consistent with your 3D map. The 3D map might still be wrong. It shows the consistency. But one thing that helps to make you more confident is when you also look at the angle distribution, you see that all the angles are covered. Now, if all the angles are covered and your projections are consistent with the 3D, you can be a little bit confident that this is actually a decent 3D reconstruction. But it's not a proof. So I would say you should also, you only check the consistency between 3D and 2D, and you should also check the angle distribution. Tilt pair analysis was a very nice way that was introduced to actually look at it. I'm just going to go through it very quickly. You took your images at 0 degree, but in the end you took a few tilt pairs, where you took an image at say -5 and +5 degree. And then you got your particles picked, the same particles from both of those picked. And so you now have your density map that you obtained with the untilted data, and now you have these tilt pairs, just 1 or 2 images, so 20 or maybe 50 particles, and you align these to your density map, and you assign the matching projections, so you know what the rotation parameters are for these projections. And the difference between those should actually be consistent to the difference in tilt that you took, because you know that. So the only difference between these should be a 10 degree rotation. And so you do a plot where you plot the tilt direction against the tilt angle, and if everything starts to cluster in one area, which should be your tilt angle, which in this case was by 40 degree, you're actually fairly hopeful that this is correct model. We can just skip over this. So this was so useful that the EM database actually now gives you this tool. So if you deposit a structure, you can actually look at this. You can give your input data. But again, these days, when you get sub-nanometer resolution, you can see the pictures of the helices. All this is really not that necessary anymore. This was only at a time when you really didn't know whether the blob that you had was correct or not. Gold standard FSC is certainly okay, but I don't think if you know what you're doing, you can get the correct resolution without the gold standard. Randomized phases we're going to skip because nobody does it. It's a very good tool, but nobody does it. And then I feel that appearance of expected secondary structures is a very important criteria. So especially these maps, which were at the edge at the time, just getting to about 10 angstrom resolution, you started to see something that looked a little bit like a helix, so maybe you actually have the resolution. But if you don't see any of this, if this is just one solid density, then you definitely don't have 10 angstrom. And so what I would say, whenever you have a structure, even at high resolution structure, the best thing you can do is to evaluate it with respect to what is known about it. So we did this, for example, with this APC structure. We had a lot of antibodies, so we did a lot of antibody binding, so we could roughly say what the subunits are, and we could compare this with a interacting map that was obtained by used to hybrid. So it actually matched up pretty nicely, so we were fairly confident that this was a reasonably accurate structure at the time. And the best way, again, this is the olden times when you had blobs. A good way to analyze your density map was if you had structures of domains or subunits of a complex, and you can dock them in and it actually fits. And so at the time we were working on the AMPA receptor, and there were crystal structures for the N-terminal domain, for the ligand binding domain, and the transmembrane domain is similar to the KCSA potassium channel. So we did a 3D reconstruction, and all this fit quite nicely. We were actually missing some density, some alpha helices, so it was actually fitting quite well. But it's a very low resolution map, but the crystal structures fit, so that is actually good. But we took a lot of criticism for the structure because at the time this was a Cryo-EM structure of the AMPA receptor. Everybody said, well, this is Cryo-EM, this must be correct, this high resolution, but there's no way to dock any of the crystal structures in there. But our negative stain structure was much lower resolution, but all the density, all the crystal structures fit very easily. And in the end, when they got the crystal structure of the AMPA receptor, it actually confirmed what we had. Our density map was actually quite accurate. This was actually unfortunate deformation by adsorption to the grid, but some of the averages looked really completely identical to the crystal structure. So at the time when you really just had these kind of blobs and you didn't know which one is accurate, docking crystal structure was a very good way to figure out what you have. And so getting back to this, so this is just the most famous example because you have 4 structures completely different of the same protein. But it was very, very common that you had at least 2 or 3 structures for different protein, for the same protein. And so this had a very storied history. So in 2011, a structure came out at 11 angstroms, just above a resolution where you would be able to see alpha helices. So again, you have blobs, and are these correct blobs or not? But because there was so much history in this, they did a very good job looking at all the different validation techniques. So they first figured out that they actually can see alpha helices, so it is close to 10 angstrom. It had the expected secondary structure. They compared their projections from the map with references that were either supervised or non-supervised, and then the particles. So it looked pretty good, that the averages looked like the reprojections. They did the tilt test, and that was actually really fun. So this is the new structure, and these are the old 3 structures, and they're just total random. So these density maps really had nothing to do with the correct structure. And then what they also did, what we also sometimes did, was use different programs to get the density map. The same data set, but use different, and they looked all very similar, so that helps you be more confident, because they all use slightly different approaches. And then the real proof came in 2015, where this structure came out at 4.7 angstrom. It's a massive, massive protein, so you have a lot of information in that. And so this is again 4 years old, 3 or 4 years old. And last year, the latest structure came out, this time now at 3.5 angstrom from Rich Hyde, who gave a lecture here. This is really a paper I really like because it shows a lot what you can do these days with electron microscopy. You're not just going to get a high resolution structure, but by binding a ligand, in this case IP3, you can actually see that you have 2 different types of how IP3 is bound to the receptor. And these 2 different types can actually occur in the same tetrameric channel. So you can now really analyze these proteins individually inside of the oligomer. And then you can also do a different ligand, in this case calcium. You see the total opening of the X-membraneous domain. And based on all this data, it was possible to really build a nice mechanistic model how IP3 receptor will be activated. So EM has come a long way from docking to from blobs to these kind of really amazing 3D structures. And because we now usually get to these kind of resolutions, map validation is done essentially by model building. So that's why I didn't talk so much about it, because it's ancient history this time. Questions? How many minutes do I left?
-3.
You're
No, that's
evil.
great.
You're
Yeah.
evil.
I only have one question from the YouTube channel, and this was back when you were talking about the cross-linking and looking at the different native versus the cross-link structure. We have Lolita who's asking, will NMR be applicable for discriminating cross-link from native state if they are mixed together for some reason? Could you use NMR to separate those 2 structures if they were mixed together?
NMR.
Yeah. I'm not sure why she's asking you an NMR question, but I think
No,
it's just like a general question.
Yeah, I still don't quite understand the question, I have to admit.
I think she's asking if you were to mix two different samples, right, like cross-linked and native state
Mhm.
without cross-linking, could NMR separate those 2 structures? But
My NMR knowledge is too limited to
Yeah.
answer
I
this.
feel like you would actually be able to just do this in single particle, just classify out if you were to mix two samples. I don't know why you would, but you could in silico. So.
So yeah, I don't. So actually, when you cross-link a few parts of the protein.
Mm-hmm.
And what Brian Scheid has done at Rockefeller with Mike Rout on the nuclear power complex, they actually found that you only have to do a few cross-links to stabilize the entire nuclear power complex. So even when you do the mass spec analysis of cross-linked samples and you find all the different cross-links, these cross-links, only a few of them actually occur in each protein.
Mm-hmm.
So the cross-linking, even if you cross-link, there might be proteins that are totally non-cross-linked and some that are heavily cross-linked. There's a mixture of all kinds of difference. So if you cross-link, you don't end up with one single species. It's still different species,
Okay.
more or less heavily cross-linked.
Okay. Beyond that, any more questions for our speaker? All right. Well,
Apologies again for being
no.
late.
Thank you very much. So I think this week there's no practical on Wednesday, so no class on Wednesday, and then next Monday we continue with Kathy Lawson. So she'll be talking about more validation, data archiving, things like that.
That's validation of the model, of the atomic model. That's why I don't talk about it.
Yeah. No, this was great. I think we didn't get to go this much into detail about every single step coming up to the
More
value, so.
detail I was hoping. I was hoping this was just an overview.
No, this was great. Yeah.